Hello, everyone. My name is JiaLi from the University of Duisburg-Essen. Today, I'm going to share with you reducing exosome secretion leads to an increased accumulation of subviral particles and HBV virions, but limits the export of autophagosome-associated HBV components. Recently, we find that endosome and autophagic pathway coordinate HBV secretion. Under normal conditions, HBV virions and subviral particles can be released downward through exosomes by the endosome pathway. But under certain conditions, such as ER stress, HBV virions and subviral particles can be recruited to autophagosomes and released. The formation of MVBs from early endosome is partly regulated by neutral sphingomyelinase, which can be inhibited by JiaLi-2022HBV, and the fusion of MVBs with the cell plasma membrane is regulated by RB27. So we came up with our questions. How do the interference with the infection of late endosomes and exosome secretion affect HBV traffic and release? Do amphisomes serve as a conventional secretion pathway for HBV? And here is the workflow. And we also got some results. Firstly, we find that JiaLi-2022HBV treatment blocks HBV virion secretion and sequesters subviral particles and virions within cells. Firstly, we treat the cells only for 4 hours. We can see it only slightly increased the intracellular HBV DNA levels, but it significantly blocked the HBV DNA level in the supernatant. However, after a non-treatment of JiaLi-2022HBV, we can see it significantly increased the level of intracellular HBV DNA, while it significantly blocked the secreted HBV DNA level. At the same time, we also see the intracellular and secreted antigen also increased, and the HBV kept the same. As we also find the accumulated HBV virions within cells. So next we detect the ER stress, and unsurprisingly, we find that the ER stress increased after GW treatment, and the colocalization of surface antigen and the PDI increased. We also find after GW treatment, the phosphorylated level of AKT, mTOR, P70, S6K, and total level of ULK1 decreased, but the phosphorylated level of ULK1 increased. And then we detected the association of amphisome and surface antigen. And very interestingly, we find that there is a structure. Here we can see surface antigen positive, LC3 positive, and the CD63 positive structure, which represent amphisomes with HBV components. And this structure can be increased after GW treatment, both in HU7 cells and HepG2-15 cells. And finally, we focused on RB27. And we can see here, after the 27A and 27B knockdown, surface antigen appeared to be perinuclear accumulated, and the HBV DNA and the surface antigen level in the supernatants were decreased, and that of intracellular HBV DNA increased after RB27A silenced. So here comes the conclusion. Firstly, we think HBV release requires the autophagosome and MVB axis, and then silencing RB27A and B, the two proteins also required for exosome release through MVBs, significantly reduced HBV release from HBV producing cells. Well, that's about my reports. I'm very happy to take your questions you may have. Thank you.